All right, looks like we're recording. So just so everybody knows, don't chat really weird things. It'll be recorded in perpetuity. So without further ado, it's my great pleasure to introduce Katrina Twing for the first network speaker series of the 2016 session. I'm Beth Orcutt, for those of you who don't know me. I'm one of the C-DEBI senior scientists, and I had nominated Katrina to give today's seminar. I had the pleasure of sailing with Katrina last fall on the expedition to the Atlantis Massif to study microbes involved in serpentinization in the subsurface. And this was really a continuation of the work that Katrina has done in her PhD and now into her postdoc at the University of Utah. So she's going to be talking to us quite a bit about the work that she did during her PhD at the Coast Range ophiolites, studying serpentinization and the microbes involved in that process. And I really look forward to it. If you guys have questions while we're going, you can type them into the chat box, and there'll be a session for question and answer at the end. And just in case you don't have this on your calendar already, the next network speaker series will be on April 28th, featuring Olivia from the University of Hawaii. So make sure you get that on your calendars. And without further ado, we'll let Katrina take over. Thanks for joining.
Great. Thanks, Beth. All right. Can I just get some confirmation that you can see my slides and hear me? Yeah. Perfect. Great. All right. Well, thank you very much for that introduction, Beth. And thank you to C-DEBI for this opportunity to share my research with you today and to everyone who is tuned in here. Like Beth said, I studied the serpentinite environment and the microbes associated with it for my PhD, which I completed this past fall at Michigan State University with Matt Frank. And that is the research that I'll share with you right now. So just a quick introduction to what serpentinization is. It's a water-rock reaction involving the mineral olivine. And in the presence of water, olivine from the ocean crust gets converted into serpentine. And when this happens, it releases copious amounts of hydrogen, which can interact with carbon in the system to abiotically form methane and small chain organic molecules. This process also results in very high pH to the system, which can be challenging for life. And so there are numerous sites of serpentinization kind of sprinkled around the world. The process starts in the ocean crust in most cases, and then the ocean crust can become abducted onto continental crust, which gives us some access to this deep sea crust a little bit more easily than going out to the middle of the ocean. But the most well-studied site of serpentinization is the Lost City hydrothermal field, which was discovered by University of Washington scientists in 2000. And the Lost City hydrothermal field is, the waters there are high pH, between pH 9 and 11, high temperature between 40 and 90 degrees Celsius, and there's an abundance of hydrogen and methane coming from the seeps. And microbiological studies of the chimneys at the Lost City hydrothermal field have indicated that the chimney interiors are dominated by really a single phylum of methane cycling archaea, whereas the chimney exteriors, which are more in contact with the fluids, or with the ocean fluids, also contain some methanotrophic bacteria. So in addition to the Lost City, as I mentioned, there are sites of serpentinization in continental settings, and this is just a map of some of the sites that our research group and collaborators have been studying over the last few years. One such site is the Tablelands ophiolite in Newfoundland, Canada, where these these pools, which are influenced by serpentinization, collect waters that can be collected. And so in the subsurface, serpentinization is taking place. The water transits through an unknown path up to the surface, and then there's a little spring where pH 12.5 water comes out and collects in these pools. And so by looking at the microbes in the system, we can get a feel for both who's living there and what sort of environmental parameters might be correlating with or affecting their life there. And so in one of these pH 12 pools at the tablelands, very low diversity was found looking at the 16S rRNA gene. The fluids were dominated by two taxa, Hydrogenophaga, who are members of the beta proteobacteria, as well as some members of the firmicutes. And through a network correlation analyses, the researchers were able to determine what environmental parameters correlated with which of these taxa, and they found that obviously the very high pH and highly reducing fluids were correlated with the presence of these organisms. And so from both that research at the tablelands as well as some other research that our group and other groups have done around the world in continental serpentinites, Matt and Billy developed this model for what might be going on in the serpentinite subsurface environment. And so down at the end member fluids, we have serpentinization, which is creating hydrogen and methane and other electron donors and organic carbon species. And down at depth, we see a lot of these anaerobes, so the Clostridia or firmicutes that I pointed out at the tablelands. And then as that water transits up to an area where there is more oxygen, as it becomes oxygenated closer to the surface, we see aerobes or microaerophilic bacteria, such as the beta proteobacterium Hydrogenophaga. And so that's kind of our working model of what is going on within the serpentinite subsurface environment. Some additional research has been done at other sites, including the Cedars in Northern California, that suggests that the candidate phylum OD1 as well as Chloroflexi are also found at these more end member fluids. But one thing that I think is really important to keep in mind here is that many of these sites were studying fluids that have come up to the surface, and we're inferring what's going on in the deep subsurface. And so I think it's very important to understand that the subsurface environment is not just empty space where fluids and bacteria are. It's a rock-hosted matrix. And so I think to really understand what's going on in the subsurface environment, we need to not only think about fluids that we can sample easily, but also about the rocks and hard substrates that are down there. And so to try to address that a little bit more completely, the Coast Range Ophiolite Microbial Observatory was established in 2011 in Northern California with funding from the NASA NIA. And so Chromo is located about a 3-hour drive north of San Francisco, which is made for good sampling trips around AGU time every year. And it consists of two main sets of wells, so that are located about 1.4 kilometers from each other. We've got the Quarry Valley well and the Coreshed well. And in addition to those two main wells, we've also drilled a set of monitoring wells surrounding them to varying depths and much to our surprise and happiness, to varying different geochemical gradients, which has allowed us to assess microbial diversity among these different gradients in the field. And so here's just a picture of the drilling process. I should also note that since this study was set up to specifically look at microorganisms, we made sure to do the drilling process as cleanly as possible, and sterile ozonated water was brought in to use as drilling fluids as part of this process. And so today, what I'll talk to you about are two sets of findings from Chromo. I focused on both the fluids that come from the wells. So since the establishment of Chromo in August 2011, we've sampled almost quarterly every year to collect samples and see who's living there. But I'll also discuss the cores that we obtained from a couple of those wells during the drilling. So first, I will start off by describing my fluid results to you. So as I mentioned, we have seven different wells that make up Chromo. Our two main wells, so CSW11 here, which has a pH of 12.2, and here is QV11, which has a pH of 11.5, and then they're surrounded by monitoring wells adjacent to them, drilled to different depths. And so, as you can see from this kind of busy table, there is a variety of different geochemical gradients, including pH here. And so our pHs range from the highest pH being CSW11 at 12.2, down to a couple of these wells that are more circumneutral, CSW14 and QV12, which are about 7.9. And so one thing that we wanted to look at this study, similar to the study that was done at the tablelands, is not only the microbial diversity, but to also kind of get at what might be driving microbial diversity in this system and how the environment and the microbes interact with each other. And so just to start out with, I did a 16S rRNA gene a study of the site. And so to orient you, we've got our high pH samples here at the top, CSW11 with a pH of 12.2, followed by QV11, and then coming down to our more circumneutral samples. And I just want to point out that these data represent operational taxonomic units, which were organized at the 97% sequence similarity level, but that each color that you're seeing here is an individual OTU. A lot of times when data like this are presented, where OTUs may have been made, they then get collapsed down into kind of higher taxonomic groupings to be able to visualize the data. The reason why I'm pointing this out is because it's very striking how low diversity is in this system. In particular, with this most extreme site, most extreme well, the CSW11, you can see that between 50 and 60% of the samples are made up of a single operational taxonomic unit, which has been classified as the beta proteobacterium belonging to the family Comomonidaceae. And spoiler alert, it's very closely related to the ones that we saw at the tablelands. And likewise, when you get down to this next extreme site, the QV11, you still have that single OTU of beta proteobacteria, but it's replaced with a handful of OTUs of firmicutes or Clostridia. And I probably should use my slide to help me share that information with you. And so once again, these samples, especially the most extreme, are dominated by beta proteobacteria and firmicutes, like we've seen before. And then as we get down to kind of the more moderate pHs, so pH 10 and below, we see greater diversity in the samples. Although we still do see a lot of the beta proteobacteria and Clostridia. And so just to focus in on this single operational taxonomic unit of the Comomonidaceae, so again, this is a member of the beta proteobacteria, it makes up between 50 and 60% of our most extreme sample, and it's still present even in our lower pH wells, but at a much lower abundance, making up maybe 5 or 10%. However, if we add in the rest of the OTUs that classified as beta proteobacteria, you can see that there's still an overall about 50% of the communities, even at the lower pH, are beta proteobacteria. The diversity just shifts from that single OTU to a couple of other ones. And likewise with the firmicutes here, you can see we've got two main firmicutes showing up in our most extreme well. There's a greater diversity as well as a greater abundance of firmicutes in QV11, and then firmicutes show up throughout the rest of the samples except for the lowest pH sample, CSW14. So very low diversity there. So don't be frightened by this figure. I'm mainly showing it to you just to illustrate how much data there is and how many different things there are to look at here, and then we'll zero in on what I think are some of the more important points. So this is a correlation network diagram, and each line that you see here is a correlation between environmental parameters and each other and OTUs. And so the operational taxonomic units are represented with these round nodes, and the size of them is relative to their abundance in the sample. So this really big one right here, which we'll focus on in a moment, is that Kamamona-DCA that makes up about 60% of one of our samples. And so as you can see, the network diagrams can help you visualize really complex data and begin to tease apart relationships that might not be so obvious. And so to try to zero in a little bit, I ran statistical analyses to determine what environmental parameters had the biggest correlation with the community composition of the samples. And a best analysis indicated that pH, carbon monoxide concentration, and methane concentration had the highest correlations with community composition as a whole. And so I zeroed in just on those three parameters and their associated operational taxonomic units here to see what was really driving these communities and their members. And so again, these round nodes are OTUs, and they are color-coded based on their class or phylum. The green lines are negative correlations, and the blue lines are positive correlations here. And so while we didn't really need to have a network diagram tell us that the two samples that were most abundant in our high pH well are positively correlated with pH, this sort of network does give us some other information. So I want to stress, first of all, that a correlation does not mean that the microbes are either producing or using these chemicals, and I think that that's important right here with the methane. There are a lot of OTUs that are correlated with methane that are not known to utilize methane, right? So we have some firmicutes here and Bacteroidetes and other groups where methanotrophs are not known. But I think what a network diagram can let us see are kind of what are important potential proxies within the environment, and carbon monoxide, methane, and pH are good proxies for the serpentinite environment based on this analysis. And so just to highlight our most abundant OTUs here, this OTU1, which was classified as a beta proteobacterium family Kamamona-DCA, it shares 100% sequence identity to serpentin amonis B1, which is an isolate from the Cedars, a nearby serpentinite site. And then this OTU over here, which was very abundant in our QV11 samples, and I think it was classified as Clostridia, it also shares 100% sequence identity with Dithiobacter, which have both been isolated from chromo and microcosm samples that we've done, but also from a serpentinizing site in Portugal. And so now that we've looked at the diversity and the geochemistry, I really wanted to dig into the metabolic potential in the serpentinite subsurface environment. So we have metagenomes from 4 of the sites ranging in pH, again, from 12.2 down to pH 8. And I looked for target genes that were indicative of different metabolic pathways. And so the first thing to note right here is that we don't see any genes for methanotrophy in our highest pH wells, and we only see a small abundance of them in our more moderate and low pH wells. Similar to findings in the tablelands, there's an abundance of both nickel iron, but particularly iron hydrogenase genes in the high pH wells. We found genes involved in aerobic carbon monoxide oxidation with the COX-DEL gene and anaerobic carbon monoxide oxidation with the CoS gene, and both of those were found in abundance in our high pH wells, as well as genes, the C-DEBI gene, so a gene in carbon fixation by the Calvin-Benson-Bassam cycle. And I should note that serpentin amonis is known to contain the C-DEBI gene, and that microbe is very abundant in our samples. And so just to kind of reiterate what we've gone through here, the most extreme pH wells, so those ranging between 11 and 12.2, were dominated by a single operational taxonomic unit of beta proteobacteria and a couple of OTUs of Clostridia. And this is really consistent with what we saw both in the tablelands and then what was developed in that model. The more moderate wells, so those with a pH between 8 and 11, showed greater diversity. And while they still contained beta proteobacteria, they had other beta proteobacteria present than just that single OTU. And I should mention here that I didn't intentionally leave out archaea in this study. They were not detected. So we tried to sequence for archaea and the 16S rRNA gene for all of these samples, and they were not able to be sequenced. I looked for them using quantitative PCR, and they were at very low abundance. And furthermore, in the metagenomes, only 1% or less of genes within each of the metagenomes was classified for archaea. So unlike other sites of serpentinization where they have seen some URI archaea, there are not archaea in the chromo fluids. So community composition is strongly correlated with the three parameters pH, carbon monoxide concentration, and methane concentration. And as far as the metabolic potential, genes for aerobic methane oxidation were found in the moderate wells, and they were completely absent in the higher pH wells. I also looked for the MCRA gene, so indicative of either archaeal methanogenesis or methanotrophy, and not surprisingly, it was not detected in any of the metagenomes. Genes for aerobic anaerobic CO oxidation, were found in QV11 with a pH of 11.5, and genes for CO2 fixation, consistent with the presence of serpentin ammonis, were found in CSW11 with a pH of 12.2. So to shift gears a little bit more, now that we've addressed what we see in the serpentinite fluids, I'd like to talk to you about what we see in serpentinite cores. So before we get too deeply into the core data, I want to tell you a little bit about the methods I used to analyze them and kind of give you some background for why we felt that going this route was important. And so I'm just going to talk a little bit about sequence data and how sequence data are generally dealt with. So we have our sample of interest. In this case, it's very low biomass, precious subsurface sample. And then in order to be confident of what microbes are actually present in that sample, we take a lot of background or blank samples as well. And generally what happens with sequence data is you've got your sample of interest, you've got your background or your blank sample, and then any sequences that show up in both of them are considered contaminants. And it goes under the premise that anything that is found in both likely came from the background, and we might as well throw it away and make sure that we don't include it in our sample since we don't actually know where it came from. But I would argue that this might be a little bit too conservative in that, so anything that comes in from either one of these samples is now removed, and it's just as possible, one might say, that the quote-unquote contamination could have came from your sample into your background sample, and I'll give you some data about that here. So this is just some data from one of our sites in Italy. This is the Ligurian ophiolite in northern Italy, where up here we've got, so again, subsurface serpentinization is taking place. The fluids travel up here to this little hole where we've got pH 12 fluids coming out and collecting in a pool. And then below us is a river with pH 9 water, probably because it's got serpentinization fluids feeding into it, increasing that pH. And so we collect both our sample of interest and background samples to try to get at who is really, who are the real inhabitants of the high pH sample. And so here, these are archaeal data And I should note that here we're talking about individual unique sequences, not OTUs. So we have a single unique sequence that shows up about 16,000 times in this pH9 river. That identical sequence shows up 200 times in our sample up here. And so it's understandable, and I would say probably correct to say that it's very likely that it's a true inhabitant of this river and it contaminated our deep sample somehow, whether that's during handling or just naturally by being near the same environment. And so we would be justified in removing this sequence from our sample of interest to make sure that it's not included as a contaminant. However, what about in this instance where we have this methanogen sequence? We have 17,000, there are 17,000 indications of this unique sequence up here in our high pH sample, and there are 20 copies of that exact same sequence down here in our background river. Now, using this conservative method, because it shows up in both samples, we would have to throw it away and say that it's clearly a contaminant and likely coming from the river. But that doesn't seem realistic in this case. So instead of just deleting things that were found in both samples, we decided to take a quantitative approach to determining what sequences statistically were likely to come from which one of the samples. And so this approach is based on differential expression, which is used in a lot of RNA studies. And so what we have here, each one of these dots over here in this plot represent unique sequences. And the ones that are colored in red were found to be statistically more abundant in one type of sample versus the other. And so we've got a handful of sequences that were statistically more abundant in the background river, including that MG1 sequence that we talked about before. And there were a lot of sequences that were statistically more abundant in the background, or pardon me, in our high pH spring, including that methanogen. And so here we can use a statistical way to determine what sequences are significant to each one of these sites, as opposed to just throwing away anything that is in both of them. And so I employed this method to determine who are the actual microbes living on the serpentinite cores. And so what I'm interested in are the microbes living here in this red box, so actually on the cores. And I used kind of two different background samples, if you will, the serpentinite fluids, and then soil samples that were cored from shallow cores up above the wells. And so it's just to give you a little bit of workflow and kind of how our collaboration has worked. Chromo is a very big project with lots of collaborators. And to make sure that we were working on the same piece of rock, when the cores came up and were split, a piece of each core went to each of our collaborators. And so mineralogy was done by Don Cardes at URI and Tom McCollum at UC Boulder. And then samples for chemistry went to Tori Holler's laboratory at NASA Ames. And a piece of the rock came to Matt Schrenk's lab, where I looked at the microbiology. And so for these samples, I looked at the 16S rRNA gene to look at the diversity. And so just a little bit of information about the cores here. So I'm not sure I mentioned, we only have cores from the two extreme wells, so from QV11 and CSW11. The monitoring wells were not drilled in a way that produced core samples. And so here is the lithostratigraphy of the well. And so the first thing that I really just want to point out here is that the cores are fairly heterogeneous. And so we don't have like clear zones of different minerals. It kind of switches back and forth throughout the depth of the core. And right here is a XRD image of one of the deeper parts of the core coming from about 41 meters here, indicating the serpentine and magnetite in there. And then along with the lithostratigraphy, we also had different measures of life and organic chemistry. And so here we have organic carbon and nitrogen throughout the well at different depths. Right here I did quantitative PCR on the 16S rRNA gene, targeting both bacteria and archaea. And then this last panel here is showing the amount of DNA that I was able to extract from the different cores. And so the first point that I want to make, this line in that last panel represents the limit of detection for the fluorometric method that I was using to quantify my DNA. And you'll notice that a lot of the samples fell below that, that limit of detection. However, the open circles represent samples that, while I did get DNA from them, they were not able to be sequenced. So they were sent off to MBL for sequencing, and they failed for one reason or another. And the filled in circles and squares represent samples that we were able to get sequences from. And so back to the DNA concentration, I just want to show that even though samples were not able to be quantified and clearly had very low amounts of DNA, in many cases we were able to sequence the DNA that was found in those samples. And also kind of note here is, you notice that at the surface we've got kind of higher DNA yields and 16S copies, and then as a rule it tends to decrease as we get deeper in the sample, with the exception of this one core around 30 meters where we saw a peak in organic carbon as well as 16S copies and DNA yield. So this way we can get a full profile of molecules associated with life throughout the well. So these data are using that differential expression technique that I mentioned before. So as I said, I had three main sample types. There were serpentinite soils, which were taken at the surface near each of the wells. There are also fluids, the same fluids that I discussed in the previous section, and then we have the serpentinite cores that were able to yield sequences. And so I treated each one of these groups as separate samples for the differential expression method. And the first finding was that there were bacteria that were determined to be fluid specific. So they were statistically more enriched in fluids than in any other samples. And this was nice confirmation. It's both the beta proteobacteria as well as the Clostridia that we've seen throughout the fluids before, and they were found in similar abundances to the fluid-specific analyses done prior. I should note that while not many people have studied serpentinite rocks before, because serpentinization creates these particular rocks, and then those rocks get weathered into soil, and these soils have very different parameters than normal soils. They tend to be high pH, which is not, which would be expected with the pH of both the rocks and the fluids, and they tend to be high in different chemicals. And so the result is that the serpentinite environment on a macro level has very specific plants and animals associated with it. And because of these very specific plants that can grow only in the serpentinite environment, microbiologists in the past have studied serpentine soils. They just haven't gotten deeper than the rhizosphere. And so the reason why I point that out is because when I did this differential expression method to determine what taxa are specific to which types of these samples, the taxa that came up as specific to the serpentine soils are ones that have been described in the literature previously as being found in serpentine soils. And that leaves us with our core. So with those two pieces of information, both the fluid specific and the serpentine soil specific taxa tell us is that now we can look at who are actually associated with the cores. And I should note that if you look down here at the bottom axis, we're talking about percent of the total bacterial community. And so a lot of these for the soils, there may be 3 or 5% of the total community, and the core specific taxa are maybe between 5 and 15%. And I just want to point out that the remainder of those communities were taxa that were not able to be determined as specific to one sample type or the other. So we can be rather confident that these taxa are actually found on the rocks and not elsewhere. And so just to kind of highlight some of the bacteria, again, there was only between, maybe 5 and 15% of the total bacterial community was determined as being core specific. And I just wanted to highlight a couple of those members. So one thing that I noticed is that there were members, both of the delta proteobacteria and beta proteobacteria, who are associated with nitrite cycling. And so here in this table, I only classified them down to the class level, but the taxa that I saw belonged to the Nitrosomanodaceae and Nitrospinaceae families. So we see microbes associated with the cores that might be involved in nitrite oxidation as well as ammonium oxidation. And these are pathways that we don't really see in the fluid samples. Additionally, 2 taxa that I want to highlight here are the candidate phylum OD1 and Chloroflexi. And so if you recall back to the introduction, I mentioned that the group at the Cedars had suggested that these 2 taxa as well as the Clostridia are really the end member serpentinite fluid inhabitants. And we did not see them in any of our serpentinite fluids. However, they were statistically enriched on the cores in the serpentinite subsurface environment here. And so we were able to find archaea on the cores. So again, we looked for archaea in the fluid samples, and they were not detected either by quantitative PCR or through sequencing analysis. So that's why we do not have a fluid-specific community here. But we do have archaea in both the soils and in some of the cores. And so in the case of the soils, they made up between 25 and 40% of the communities, and they're exclusively from the soil crenarchaeodal group. And then in the case of the cores, save for this one, between 40 and 50% of those communities were determined as being core-enriched archaeal taxa. And so again, here's a table of the core-enriched archaeal taxa, and we have finally found the methanogenic archaea that had been hypothesized to exist in the serpentinite subsurface environment in that initial model, but were not found in the serpentinite fluids at Chromo. And so likewise, to the fluid analyses, in addition to doing 16S rRNA analyses on the samples, I also wanted to try to correlate the different taxa that we were seeing back to some of the environmental parameters, including mineralogy. And so here is a pie chart of the percent of archaeal sequences that were correlated with different mineralogies. And so first of all, you can see that 60% of the sequences were not able to be correlated to any lithostratigraphic or mineralogical data. However, about 39% of the sequences were, and so about 2% of the sequences were positively correlated with, I'm sorry, that got cut off, detrital serpentinite soils, and about 37% of the archaeal sequences were positively correlated with the magnetite-bearing serpentine, which is found throughout the core, and of which this XRD slice down here is from. And so of those 37% of archaeal sequences that are positively correlated with magnetite-bearing serpentine, about a third of them are methanogens. Another third are from the miscellaneous crenarchaeodal group, and then the rest are dispersed through other archaeal taxa. And so just to reiterate what we found with our serpentinite cores, so we were able to identify core-enriched taxa within the serpentinite cores. With regards to the bacteria, we see nitrite oxidizing and ammonia oxidizing bacteria, which had not been present in the fluids. And we also see the OD1 and chloroflexi, which previous studies had suggested were endemic to end member fluids. But these analyses suggest that they might be actually endemic to the solid surfaces themselves. And we were also able to find archaea, which were not present in fluids at Chromo, including methanogens, which are strongly associated with magnetite-bearing serpentine layers, as well as MCGs and some SAGMEGs, which have been found in other deep samples with high pH and high methane concentrations. And so just to bring it back to that model that we looked at in the beginning, we have serpentinization creating hydrogen and electron donors and organic carbon and transiting up to the surface. Consistent with the model, particularly in our very high pH fluids, we found an abundance of both anaerobic Clostridia as well as these aerobic beta proteobacteria, whether you want to call them hydrogenophaga or serpentininomonas, they have 100% sequence identity to each other. Additionally, we found methanogenic archaea and OD1 and chloroflexi associated with cores, as opposed to being kind of free living in the fluid state. And so this kind of, it reinforces what we had predicted was happening in the system. But I think by looking at it on a greater scale and across different geochemical gradients and also different sample types, we can really begin to know what's happening in the subsurface environment here, as opposed to speculate from a handful of surface samples. And so just to summarize what we found again, so in the fluids, we have very, very low diversity, dominated by beta proteobacteria and Clostridia. The parameters pH, carbon monoxide, and methane concentrations were strongly correlated with community composition. Methane oxidation genes were only found in the moderate pH wells, so those are wells between pHs 8 and 11, I guess 8 and 10, sorry. And CO oxidation and CO2 fixation genes were found in the high pH wells. In the cores, we were able to identify core-enriched taxa, including diverse bacteria and members that are involved in nitrogen and ammonia cycling. We have bacteria that were thought to be from end-member serpentinite fluids were found to be core-associated, and we found archaea, including methanogens. And for future directions, I just want to reiterate again that this is a very large interdisciplinary ongoing project. And so there are a couple of different directions that we could go. And I think that one thing that will really help kind of bring this all together that's currently being investigated is the hydrology and connectivity of the wells and the fluids in the subsurface. Another direction that this research could go, and I think would really benefit from, is looking at microbe-mineral interactions on a finer scale. I was able to begin to assess that, I think, with the correlations in the core project, but I think now that we have a couple of targets, such as magnetite, we can really focus in on that a little bit more. And again, anything that we've learned from this initial serpentinite drilling project at Chromo, I think will be valuable going ahead and ongoing drilling projects, both in Oman and also with the Atlantis Massif drill project that Beth mentioned earlier. So with that, again, I would like to thank C-DEBI for the opportunity to share my research with you today. I'd like to thank my PhD advisor, Matt Schrenk, and his past and present lab members at Michigan State University. I'd like to thank my postdoc advisor, William Brazelton, as well as his lab members, specifically Chris Thornton and Alex Hare, who have been invaluable in assisting me with some of the bioinformatic analyses. Thank you to my Coast Range ophiolite collaborators, and also to the Census of Deep Life and JGI for sequencing opportunities, and the DCO Deep Life community for current funding. So with that, I will take any questions.